Hi, hello everyone. So as Maidi said, I'm Audrey, representative for the CNRS in Washington, D.C. And today I'm going to introduce you the speakers for this session. So it's starting with Dan Angelescu. So Dan, if you're here, you are the CEO and Research Director of Fluidion. You hold a PhD in physics from Princeton University and HDR degree from Université Paris-Est, and your research interests are in the area of microscale physics, nanotechnology, and Fluidion initially launched in Paris, France, and now with offices also in Los Angeles, California, and you are developing, innovative sensors to monitor water system. But please feel free to talk to about us about what happened, and you have 10 minutes for that, so feel free to do it.
Wonderful. Thank you so much for the introduction, Audrey. Can everybody hear me?
Yes. Perfectly.
Okay, great. So I'm going to quickly switch. So far, we've talked about soil, about permafrost. Now we're going to talk about water and about monitoring threats and monitoring pollution in water sources. And specifically, I'm going to talk about resilience for autonomous, resilient sensors for autonomous water quality surveillance. Can we move on to the next slide, please? Thanks. So why is water quality surveillance important? It is important because it helps eliminate risks of different kinds of people being in contact with contaminated water. So that could be microbiological risks, and here we could talk about recreational water like bathing at the beach and being in contact with pathogens, or it could be drinking water. But it could be also chemical degradation of the water, which has a more long-term effect on health. And again, it's important to be able to localize the sources of pollution, and this is not always trivial, and sometimes, it's even today, it's impossible with the current technology. And to provide feedback for certain industrial operations. For example, we're looking at water reuse today. Los Angeles is looking to reuse 100% of the wastewater produced by the city. Well, that reuse requires a lot of quality control to ensure that it's safe for being reused. And why is resilience important? Well, that is important because we rely on all kinds of infrastructures, and they can fail. Communication can fail, power may not be available, so sensors are exposed to all kinds of issues. That's why a lot of people develop these networks of sensors. Even if 10% of them fail, the others will still be able to send important data. Well, when you're in water, you also need to worry about things like vandalism. People steal things when they see them. You may talk about fouling, you may talk about communication jamming and other issues like that. And of course, you can always have intentional attacks that could be terrorism or just negligence that could lead to water quality degradation. Next slide, please. So Fluidion, like Audrey presented, so we're a company. We're started in Paris, and now we're based in both Paris and Los Angeles. We're based in downtown LA, in Los Angeles. And if we can move on to the next slide. The areas we address are basically water quality in all forms. So we're trying to develop sensors that address specifically those measurements that today require a lot of resources in order to be monitored. We're looking at microbiology today to measure E. coli, for example, in a water stream. You need to send somebody out there who takes a sample, refrigerates it, brings it in a cooler to a lab, and then you only have a result like 3 days later. And that's a very long time when you're talking about people being exposed to that water, because being exposed to fecal pathogens can give you all kinds of disease. So you'd rather have something that tells you right away when the water is contaminated. So we've developed a sensor, which is called the alert system, that I'll talk about later, that does that in situ. Again, we're looking at chemical water quality, and this could be, just all kinds of chemicals in the natural environment, but it could be also chemical degradation of the drinking water. And that's an ongoing issue in Los Angeles that uses chloramine to disinfect the water today. And in the hot weather, chloramine is unstable. It develops ammonium that develops then it's metabolized by nitrifying bacteria, which develop nitrites, nitrates, and then you can have a full bacterial bloom in a water tank in the distribution network. So that requires close monitoring. And until we developed this sensor, there was nothing that could do that online. So I'll talk about that a little bit, and then I'll talk about the mobile sensor deployment. If you want to look at a reservoir, most of the time people install a sensor at one point and they think that's representative of the whole reservoir. But the reservoir is exposed to winds, to weathers. You can have cyanobacteria that develop on one side of the reservoir and see no sign of it on the other side. So there is heterogeneity, and that requires a way to do a mapping of water quality and not just measure it at one point. May we move to the next slide, please? Yeah. So what we are trying to do in Fluidion is to really use the latest technologies, and we're talking here about sensor technologies. We're talking about the newest concepts in sensors. We're talking about developing new protocols to measure either bacteria or chemicals and implementing them in highly miniaturized systems using microfluidics and eventually incorporating these technologies in sensors that are installed, like you see on the screen, really in rivers and lakes and water reservoirs that operate in the real world for months and produce data that's sent remotely to servers. So we're trying to cover that space that today is poorly covered, of water quality sensors that can be installed for long term, are resilient, and transmit data remotely and operate on battery. Let's move on to the next slide, please. And to the next again. So I'll talk first about measuring microbiology. So we've developed a technology called the alert system that essentially can do these measurements in situ. It's been qualified against laboratory standard methods, and it works really well. And now we're at the second version of the technology, which is actually manufactured in the US with our US company for the US market, and we also have manufacturing in Paris. So this is a concept that's based on a single-use or disposable cartridges where all the reactions, biochemical reactions take place, incubation takes place there, mixing with a bioreagent, and the optical measurements are performed across this optical cell. So that allows us to have a system that's very reliable, that has no cross-contamination, but at the same time that allows a very quick maintenance. You can change a cartridge in 10 seconds and you're ready to do a new set of measurements. Also, these can connect to external water quality probes that measure all the water quality parameters like, turbidity, conductivity, dissolved oxygen, pH, chlorophyll, and whatnot. So you can get all the parameters logged with just one tool. So let's move on to the next slide, please. So this shows a little bit, some of the published work on doing side-by-side comparisons between our technology and standard laboratory techniques. And if we move on to the next slide, here we see the same thing, but applied to wastewater. So we have wastewater at all kinds of treatment stages, from the primary clarification stage all the way to tertiary treatment. And you can see that the technology works well across the full concentration range in bacteria, across essentially 5 orders or 6 orders of magnitude in concentration. Let's move on, please. Thank you. This is an example from California where it's used by the regulators in California, by the California Water Quality Control Board. This is in San Diego, and we're looking at the Tijuana River that's highly polluted from the Mexican side mostly. And that pollutes the beaches near San Diego and Baja. So that's a major environmental concern. Border patrol, people are getting sick every year. Tens of people are falling sick because of contact with this contaminated water. So the California Water Board is using our sensor to monitor this kind of stream, and this is data that they produce, again, showing very good correlation with the lab. Next slide, please. This is another example of how such sensors can be used in real life. Here we see two sensors. One is near the Eiffel Tower in Paris. The other is about 2 miles upstream, and they both measure E. coli at noon every single day of the year. And they've been there for this is the 5th year that they're installed. And so we get these really, really long time series of bacterial measurements, and we can also superpose the rain events, which you see as red vertical lines. So you see that we can monitor what's called combined sewer overflows. This is where the sewage network cannot handle all the rainwater, so it has to essentially allow sewage to come untreated into the river. So we see that, and we see the effect of that. So the city of Paris is using now our sensors in preparation for the Olympic Games in 2024, where the aquatic events will be held in the river.
You have 1 minute?
1 minute?
Yeah.
Okay, so I would like to jump over the next section then, and so here, I have one more thing to say here. So these are examples of how the technology is used in the US. So we are partnering with the US Geological Survey, which is the federal agency mandated by the government with monitoring all the rivers and the environmental quality of rivers in the US. So they're using our system at multiple places now to measure E. coli. But another partner for us is the city of Los Angeles, and we are equipping the first disinfection pilot plant at the LA Sanitation Hyperion power wastewater plant, which is one of the biggest in the US and in the world, actually. And they're using our sensor in order to monitor the effectiveness of the disinfection process. So these are, this is just one example of one sensor and of different areas of application. And these kind of sensors, they produce data whether you're in the middle of a pandemic or not. So we were able to produce data throughout really difficult situations where all of the monitoring has been stopped in other places because it required people to go out and collect data. And we saw some really interesting things. For example, we saw that during the first lockdown in Paris, water quality has improved tremendously. It has essentially water quality, number of bacteria dropped by over an order of magnitude over the same period the previous years. And that was because a lot less people were actually present in Paris. Nobody worked during the first lockdown. And that really helped with the water quality as well. So anyway,
thank you then.
Thank you so much for your attention, and I'll be open to any questions.
Maybe Rebecca, you can quickly...
We'll stop the presentation right here. Thanks.